A warm welcome to my channel. The topic for today's presentation is Introduction to Operon. Now, an operon is a functioning unit of DNA containing a cluster of genes under the control of a single promoter. The cluster of genes include an operator, a promoter, a regulatory gene, one or more structural genes, and a terminator. A raw illustration of an operon is as represented. Now, an important thing to note over here is that the sequence in which these regions are represented may not be the same. Now, coming back to the operon, operator can be either switched on or it can be switched off. If the operator region is switched on, we say that the operon is on. If the operator region is switched off, we say that the operon is off. If the operon is on, transcription of the structural genes take place. If the operon is off, transcription of the structural genes is inhibited. Promoter, on the other hand, is a region to which RNA polymerase binds and initiates transcription. Now, regulatory gene can either be an enhancer or an inhibitor. An enhancer is the regulatory gene which on transcription and translation results in the production of a protein which goes and interacts with the operator region, switching it on. And if the operator is on, operon is on, resulting in the transcription of the structural genes. Inhibitor, on the other hand, when transcribed and translated, results in the production of a repressor protein. This repressor protein goes and binds to the operator region, switching it off, thereby inhibiting the transcription of the structural genes. Now, the structural gene may be one or more than one, and these are the actual genes which are transcribed in an operon. Terminator, as in a transcription unit, terminates transcription. Moreover, an operon which is regulated by an enhancer regulatory gene is a positively regulated operon. Example, trip operon, which is responsible for the synthesis of an amino acid named tryptophan. On the other hand, an operon which is regulated by an inhibitor regulatory gene or a repressor is called a negatively regulated operon. Example, lac operon, which is responsible for the metabolism of lactose. Now, if we want to inhibit the transcription in case of a positively regulated operon, we need a molecule called inhibitor. In the case of trip operon, the inhibitor is tryptophan itself. Tryptophan goes and binds to the enhancer protein in such a way that the enhancer protein is unable to interact with the operator. So, the operator is switched off and the structural genes are now inhibited from transcription. On the other hand, if we want to promote transcription in a negatively regulated operon like lac operon, we need an inducer. Inducer in the case of lac operon is lactose itself. Lactose goes and binds to the repressor protein in such a way that the repressor protein is unable to bind to the operator region. Operator remains on and the structural genes are transcribed. So this is the basic introduction to operons. The understanding of the operon is essential in order to regulate gene expression at transcriptional level. Thank you. Please like, share and subscribe for more videos.